Good.
Okay, perfect.
Right, let's go. Yeah, hi everybody. Thank you for having me. So yeah, indeed, I'll talk about testing methods for COVID-19. I work at Cancer.uk, which has nothing to do with COVID normally. But during the lockdown, yeah, things changed. So quick introduction. Usually I work on cancer on a protein called p53, which is turning here in circles, looking quite nice. It's a very important protein, basically regulates if cells survive or not in cancer. And I'm looking if the effect of metals are similar to what mutation on the genes do for that. Whatever, it does, it's just a little introduction. So, and also a little disclaimer, I am not a doctor. Well, I'm a science doctor, not a medical doctor. So I'm more specialized in the molecular side of things and not so much on patients, diagnosis, and all that kind of stuff. So bear that in mind. So yeah, the context why I'm here is basically that during the lockdown in the UK, I volunteered as a COVID-19 test person. So we had a local test center where we could go and help with the testing. Yeah, and during this and then through research here and there, I learned quite a lot of different things. And I thought I'd share that with you. So COVID-19 is the name of the disease. The name of the virus is actually SARS-CoV-2 or CoV-2 for coronavirus. And here is basically what it looks like. This is an artist rendering, this is 3D, but basically all the elements are there. It is very, very small, on the range of something like 100 nanometers. In comparison, more or less, average bacterium is 10 times bigger, 1,000 nanometers or 1 micrometer, and a human cell is 1,000 times bigger with about 100 micrometers. So yeah, really, really small. Basically, this is just a ball and a container for genetic information called RNA, which is basically similar to DNA, but it's just a bit different. It's called RNA. On the surface, you can find spike proteins. Those are the proteins or the things, and in simple terms, that attach to the human cells for the virus to get into those cells. And there's an envelope, which is basically a fat layer, which is why washing your hands with soap or with alcohol disrupts the virus, because, as you know, soap and fat, they don't like each other, and that destroys the virus. And then you have different things on the surface, the so-called envelope and membrane proteins, these little dots right there. These are electron microscope images, so these are actual images of the virus, or many of them. Usually they're black and white, so these have been colored in on the computer just to make things a bit more obvious. You can see here on the left side different sizes, so there's smaller ones and bigger ones. And on the other side, yeah, they all seem a bit more homogeneous. What's nice here is that you can see how small they are compared to the whole rest of the cell. You don't even see basically the curvature of the cell, because it's so big compared to the virus. So I told you that in the virus is a thing called RNA. We can also call it the genome, which is the assembly, the accumulation of genes that is in this virus. And basically I show you this because we'll need it a little bit later. There's different genes that are basically like an instruction manual for proteins and elements of the virus that it needs in order to produce itself. There's bigger ones like this one, medium ones like this one, and smaller ones like those last bits, for example. And all this, so these are 30,000 letters, the famous ATGC letters of DNA, very similar in RNA, and they are all packed in the virus. It's an instruction manual. Now, testing. There's different ways. There's basically two main ways how you can test for the virus. The first one is to just look for the virus, for the presence of the virus, and more specifically, specific little bits of RNA that are from the virus and that you should not have otherwise in your body. So yeah, you look at the genes. If they're there, you're very likely to have the virus. But you could also look at the response of your own body, or of somebody's body, to the virus. So basically, the immune system can detect the virus. Different cells are involved. It's very simplified here. But in the end, you have so-called B cells that produce antibodies that are here in green, and they can attack and neutralize the virus. So this is the other way. So you can look at if the virus is there, if there's the genes, or if the cell, or if your body has reacted to it. However, looking for the virus or looking for the RNA reflects on the infection that you currently have. So, once you're cured, 3 weeks later, 2 months later, or whatever, there's no virus left, you cannot find the RNA. If you look for the antibodies, they are not there at the very beginning, but they come later down the line because the body needs time to accumulate them and so on. So the antibody tests or the serological tests, they reflect on the infection that happened in the past, so more than 2 weeks actually. In order to look directly for the virus, you need to do a so-called nasopharyngeal swab, which is depicted here at the top, which is very unpleasant for those who have done it. They know what I'm talking about. I did it, and it's not nice. Basically, you sample in the back of the throat and in the nose, with a swab, and that's how samples are collected to do the so-called PCR test that we'll talk about in a minute. And if you look for the body's response, looking for the antibodies, then you just need a blood sample, and the test is called serology test. I'm sorry. We will focus mainly, or I'll focus mainly on the PCR test, because that is what is used to control the spread of the disease, of the pandemic, because it gives the immediate readout of what's happening. The antibody test is more later down the line to know who has been sick, who might be immune, how things will change with time. So as a diagnostic tool, these are not suitable. So they're very similar, these antibody tests or serological tests, very similar to pregnancy tests. This is how they work. And actually this image is from a pregnancy test illustration. So you apply your sample, here it would be the blood. In the blood would be antibodies or not from your body, and these actually would be the yellow little dots here. Then other antibodies are used, the blue ones with the little Y on top, and basically it's on some kind of paper that wicks the liquid, in this case from left to right, and at the position of these lines here, they are blue, on the bottom they are red. The antibodies can get caught while they pass, and basically indicating if there's presence or not of the antibodies in your blood, indicating if you have been infected or not. Now, let's talk about the PCR-based tests. The concept here is because you can't get all the virus that is in your body, you can only grab a little bit in your throat and stuff like that. So the idea is to amplify the DNA or the RNA so much that in the end you can actually detect it, because small amounts you can't detect. There's a little mathematical formula here, which is very simple, but just to bring it back into the math world, which is 2 to the power of n. So after several cycles of amplification, you double it, and you double it again, and you double it again. After 10 cycles of this whole reaction, this PCR reaction, you have about 1,000 copies of whatever you had before. After 20 cycles, you have 1,000,000 copies, and after 40 cycles, which is the usual length that you have 1,000,000,000,000 copies, theoretically. It doesn't go all the way that high, but to give you an idea. So you massively amplify whatever is existing in your sample that you got from a potential patient. So there's several steps involved in the whole test, in a test center. Basically, you need to receive the samples. They're all, barcode labeled and all these things, anonymized, everything. Then the virus needs to be what's called lysed, so basically broken down, open up, and extracted. Then the RNA that is in the virus needs to be taken out and isolated. Then the PCR reaction has to be prepared. Then the RNA that has just been extracted and the PCR that has been prepared need to be mixed together. Then this whole PCR needs to be run in a machine, and then the results need to be analyzed and checked for if the quality of the data is correct. And during my time in this testing facility, now I'm back working normally in the lab, I was in charge of the last bit, running the PCR, analyzing the results, and so on. So I have a few pictures of what the lab looks like. I was some colleagues that were still there sent me these images. So this is the room, one of the rooms where the researchers basically open up the virus and collect what's inside. These are sterile conditions. This is a lab where the RNA gets extracted. There's like machines that do it automatically, 96 patients at a time. Then here, this is the same room. We have a bunch of plates of reagents that are prepared to go into these machines. There's about 10 or so in this room. And then we had, in this particular lab, we had 2 rooms. Here's a room with a sterile cabinet to prepare the PCR reactions. And this basically was my realm, the machines that run the PCR. So there we had in total 6 rooms with between 6, 7, 8 machines per room. So we had in total 40 machines, and we could run about 20,000 or more samples per day. And this is just a picture of what it looks like looking at the screen. So in order to understand how it works or how it can be specific, we have to go a little bit more into detail in how PCRs work. I told you before that the DNA or RNA basically gets doubled and then doubled again. But we don't amplify the whole DNA or RNA, but just a little bit. And in order to define which bit that is, we need so-called primers. These are little bits of DNA that bind to the other DNA from the virus and basically tell us, okay, we start here and we stop there. So this is where we come back to this graph. You could test for the presence or amplify 1 bit of the DNA, but it's better to do 2 or 3. And a very common test that is used all over the world does exactly that. So there's one so-called set of primers or delineators of a region. At the beginning, there's one in the S gene for spike, actually, the virus, and then there's one towards the end. So that makes the whole test a little bit, well, or much more specific, actually. This is a zoom on the results on the computer. I have to simplify it a little bit. There's a whole bunch here, which maybe we can talk about later. But basically, the more these lines are towards the left, the more virus there is, the more positive it is. And if there's nothing, like this whole bunch of stuff here that is red, basically that's negative. And then we have some intermediate results, like these pink ones, where there's very low amounts of virus. So either the person's just beginning an infection or has just finished an infection, or something else happened. How sure can we be about all these results? So we have, of course, what it would be called the real positives. So these are samples from patients that actually have the virus and they show up as positive. So they're real positive, so we're happy. The same is true for the real negatives. Somebody has no virus, he gets tested, he turns out to be negative. Everything's good. In biology, you would refer to these to real positives. The proportion of those that indicates the sensitivity of the test, and the real negatives, they indicate the specificity of the test. But you also have what is called false positives and false negatives. So false positives are results of tests from people that are actually infected. It could be at the very beginning or at the very end, and very low levels of virus, but they are infected, but the test returns as a negative. Sorry, I just switched that. So if you're infected, but your test is negative, it's a false negative, and it actually should be a positive. And the same is true for the false positives. They're very rare. If you're actually negative, and there's no virus, and that's for sure, but you do a test, one, and that one turns out to be positive, then that's a false positive because it's not true. Here's a little, and to finish, a little visual representation. from a handy little tool that can be found online. There's a little bit of input data, so you put a pre-test probability, which is the likelihood of a person that actually has COVID-19. So that means that it is somebody's taking the test who has suspicion to actually have the virus. It's not random testing, from somebody who's completely, without any symptoms or whatever. These are mostly people that have a suspicion to be tested. So 80% estimate in this case is the likelihood to actually have the virus. Then we have a high sensitivity and high specificity. We have a lot of true positives. We have a good amount of true negatives. These are basically the other 20%, and then 1 false positives and 4 false negatives. So this one is a positive, but actually it's not. And these ones are negatives, but actually they're not. And that's a problem, because people thinking that are negative will go out, socialize, do all the things, but actually they're positive, so there's a risk that they infect people. Change the numbers a little bit, reduce the test sensitivity here, and we see that the false negative proportion is increasing kind of dramatically. 25% of people that have been tested should be positive, but think they are false negatives. Yeah, so these are theoretical numbers. And why would a test be? Why would a test have a low sensitivity? Well, it's less about the technique and less about the PCR, but more about the sampling. So in the UK, there's a lot of home test kits where people test themselves at home, and if they don't do the swab correctly, and because it's painful, it's not nice, if they do it very shortly or whatever, they might not collect enough virus for the test, and that can be a problem. Right, and I think we can talk about these things a bit more maybe in the discussions or whatever, but I'll finish with that. I hope you understood everything.